Welcome to This Explainer. Today, we're going to dive into the really fascinating high-stakes journey that a molecule has to take to prove it can survive the real world. We're looking at a foundational document in the pharmaceutical world, the ICH Harmonized Tripartite Guideline on the Stability Testing of New Drug Substances and Products. And honestly, this isn't just about mixing chemicals in a lab. It's about the exhaustive, rigorous process of proving that the medicine you take is going to do exactly what it's supposed to do every single time.
Okay, let's start with this core question. It's the whole reason this massive, highly technical global standard even exists. Think about it. When you pick up a prescription from the pharmacy and you see an expiration date 2 years from now, you probably don't think twice, right? You just trust that a pill you take on day 1 works the exact same way as a pill you take on day 700. But how do we actually know that for sure? Well, the answer is a deeply scientific gauntlet of testing.
So, here is exactly how we're going to break down this standard today. First, why stability testing matters. Second, stressing the drug substance. Third, formulating the drug product. Fourth, storage climates and containers. And finally, number 5, evaluating data for shelf life.
All right, kicking things off with Section 1, why stability testing matters.
The absolute biggest takeaway here is harmonization. The core purpose of this guideline is to unite the European community, Japan, and the United States under one single standard. Basically, stability testing provides hard evidence on how a drug's quality shifts over time when it's bombarded by temperature, humidity, and light. By agreeing on these rules, a drug that is proven stable in what we call climatic zones I and II, which covers the environmental baselines for those 3 regions, is mutually acceptable across all of them. This massive standardization is essentially what streamlines global access to new medicines.
Moving right along to Section 2, stressing a drug substance.
Okay, so here we enter the crucible with the raw material itself. We are talking about the drug substance, the unformulated active molecule. Before it ever becomes a pill or a liquid, it's just raw chemistry. And honestly, the goal of stressing this material is to intentionally break it. By forcing it to break, scientists can identify its intrinsic stability, discover exactly how it degrades, and validate that their testing equipment can actually spot those degradation products when they happen.
Now, the magic number here is 3. You can never ever rely on a single lucky run in stability testing. The guideline strictly dictates that you must test a minimum of 3 primary batches, and these batches have to be manufactured to at least a pilot scale, using the exact same synthetic route as the final product. This guarantees that the material being tested is a true, authentic representative of the commercial medicine that's going to eventually reach the public.
To find the breaking points of this raw substance, they expose the molecule to a literal gauntlet. We're talking about baking it at 10-degree increments above standard accelerated levels. They drown it in 75% relative humidity or higher. They blast it with extreme light to test for photolysis, and they test its hydrolysis across a wide range of pH values. This is how they map out exactly what environmental threats the raw molecule can and absolutely cannot survive.
Section 3, formulating the drug product.
Notice the big shift here. At this stage, we stop looking at just the intrinsic chemistry of the raw molecule, and we start testing the physical mechanics of the final drug product. This means taking the drug in its final proposed marketing container, like a blister pack or a vial, and testing totally new attributes. We're looking at things like, does a cream separate? Does a pill dissolve properly? Or, hey, does the mechanical dose actuator of an asthma inhaler actually still function after sitting in storage for months?
For the long-term testing of these products, imagine a continuous heartbeat of verification. If a drug proposes a shelf life of at least a year, we test it every 3 months during the first year, every 6 months during the second, and annually after that. At the exact same time, we run accelerated tests under higher heat and humidity to simulate long-term aging really quickly, checking in at just the 0, 3, and 6-month marks. It is a relentless, ongoing verification process.
All right, Section 4, storage climates and containers. Let's see what happens when we expose our packaged product to the real world.
It is fascinating to see how precisely these guidelines map out general case climates. To get a drug approved, developers have to submit at least 12 months of long-term baseline data at 25 degrees Celsius, plus 6 months of intense accelerated data at 40 degrees Celsius and 75% relative humidity. This dual-track system gives regulators a perfect picture of how the drug behaves normally versus how it reacts under severe thermal stress.
But what happens if the drug just can't handle that 40-degree accelerated stress test?
Well, the guideline establishes these strict, significant change tripwires. If a product loses a 5% change in its potency or if physical traits like color, caking, or hardness fail, the developer is forced to fall back and conduct intermediate testing at 30 degrees Celsius. They essentially have to backtrack to figure out exactly when and where the product broke.
Now, those rules change completely based on the packaging. If you're using a semi-permeable container, like a standard plastic bottle for a liquid medication, the biggest threat isn't just heat, it's actually evaporation. Notice how the accelerated humidity condition drops to a bone-dry level of not more than 25%. We aggressively test in low-humidity environments to ensure the liquid won't evaporate through the plastic, which would concentrate the medicine inside to potentially dangerous levels.
For medicines requiring cold storage, the whole baseline shifts. If a drug lives in the refrigerator, long-term testing is done at 5 degrees Celsius, and the accelerated test just means bringing it up to standard room temperature, which is 25 degrees. For drugs stored in freezers at -20 degrees, there isn't even a standard accelerated test. They are just exposed to elevated temperatures for short periods to simulate, what might happen if a shipping truck breaks down in transit.
And finally, Section 5, Evaluating Data for Shelf Life.
When a company applies for approval, they might not always have 3 full commercial-scale production batches that have finished years of testing. In those cases, regulatory bodies demand a stability commitment. This is essentially a binding post-approval contract. If they don't have the full commercial data up front, they are legally bound to place the first 3 production batches into long-term stability studies immediately after approval to continuously verify that shelf life.
The actual math behind determining the shelf life is incredibly rigorous. It is definitely not an educated guess. Statisticians calculate a 95% one-sided confidence limit, which is basically a mathematical guarantee accounting for any potential batch variations. The official shelf life is set at the point where this 95% confidence limit intersects the minimum acceptance criteria for the drug. This ensures there is a mathematically microscopic chance that the medicine will chemically fail before the date printed on the box. And all of this entire exhaustive journey culminates right here on the physical label. The guidelines require strict, exact wording for storage statements based entirely on the hard data collected. It completely outlaws vague, unscientific terms like ambient conditions or room temperature. The label has to provide specific, actionable storage instructions so that the product remains exactly as safe and effective as it was designed to be.
So, knowing the extreme lengths, the strict temperatures, the complex math, the brutal stress testing required to guarantee just a single shelf life date, it kind of changes how you look at your medicine cabinet, doesn't it? This framework doesn't just put dates on boxes, it fundamentally secures the entire global pharmaceutical supply chain. It ensures that whether a pill is taken in Europe, Japan, or the US, it delivers exactly what it promises. Something to chew on the next time you check an expiration date. Thanks for exploring the standard with us, and keep asking questions about the hidden systems that keep us safe.